Before going into detail physiology of microcirculation, let's first look at its brief anatomy so that we can learn what are the structures that play a role in microcirculation. We have seen in the previous slide how vessels are branching out of each other, making a tree-like structure. And we know the arterial side of that tree is made of mainly arteries. With around 4 millimeters diameter, it then branch out to arterioles, with almost 30 micrometer diameter, while the venous tree is made of veins that are around 5 millimeters diameter, and the branches that merge to form veins are the venules. Of these are obviously lesser in diameter, of around 20 micrometer. And in between these arterioles and venules, capillaries are present that are around 10 micrometer in diameter. But among all these vascular tubes, only capillaries forms the actual functional area of circulatory system. That is the exchange of materials between blood and tissues. And these capillaries are a major part of microcirculation, together with some terminal vessels. Structurally, capillaries are very narrow and short. However, quantitatively, these vessels outnumber the other blood vessels. As we know, about 10 billion capillaries are present in the body. This plenty of capillaries form interwoven networks that are referred to as capillary beds or capillary plexuses. Blood flow through these capillary plexus is generally named microcirculation. And this microcirculation is studied by focusing the capillaries of the fingernail bed of humans under dissecting microscope. So, let us have a close look at these areas involved in microcirculation. We already know arteries give rise to arterioles, and then arterioles further branch out to terminal arterioles, with even lesser diameter. From the terminal arteriole, the meta-arterioles take origin. Meta-arterioles are also called precapillary arterioles, which are short vessels directly connecting arterioles and venules at opposite ends of capillary beds. From meta-arterioles, two types of capillaries arise: preferential channels and true capillaries. Preferential channels are also called continuous capillaries. After arising from meta-arterioles, these capillaries form a network and finally join the venules. Preferential channels or continuous capillaries have the same diameter as meta-arterioles, whereas true capillaries also form a network and join the venules. But the diameter of the true capillaries is less than that of the meta-arterioles. There is another important anatomical structure to mention here. That is precapillary sphincter. The beginning of true capillaries is encircled by smooth muscle fibers. It functions as a sphincter, so it is known as a precapillary sphincter, as it controls the blood flow through true capillaries. And the relaxation and contraction of these sphincters are controlled by sympathetic nerve fibers. So, overall, all capillaries, with some terminal arterioles and venules, makes up complete anatomical pathway for microcirculation. Let's zoom this part of capillaries particularly, as it is a major part of microcirculation, and learn some of its important dimensions. Each capillary lies in very close proximity to the cells of the tissues at a distance of about 20 to 30 millimeters. This enables easy and rapid exchange of substances between blood and the tissues through interstitial fluid. Total number of capillaries in a vasculature is about 10 billion, and the surface area of all these capillaries is 500 to 700 square meter. About its size, average length of a capillary is 0.5 to 1 millimeters, and its diameter is 8 to 10 micrometer. We know that capillary is present between the venule and arteriole. So, the anatomical end closer to the artery is called the arterial end, and the one closer to the vein is called the venous end. Pressure on both these ends vary, that we will discuss later. These dimensions will later help you understand the concept of microcirculation. Now look at this image again. Here we have terminal arteriole, then capillary, and then venule. This red part from terminal arteriole to almost the mid of arteriole is actually metarteriole. And then this blue part that exists between the metarteriole and the venule is called the thoroughfare channel. This channel then joins the venule, and this venule is particularly named as postcapillary venule, which drains the capillary bed. In each capillary bed, there are usually between 10 and 100 capillaries, varying per organ or type of body tissue. Most often, they branch from the metarteriole, returning to the thoroughfare channel. However, sometimes they emerge from the terminal arteriole, emptying directly into the venule. Here is another terminology to understand, that is vascular shunt. And to explain that, I have simplified the previous image. A vascular shunt is actually the direct link between arterioles and venules, means without any capillary connection between the two. That we can see in these meta-arterioles and thoroughfare channels. This vascular shunt is also called arteriovenous shunt. And this shunt, or link between arterial and venous side of circulation, is provided mainly by meta-arteriole or preferential channels. The blood flow through this anatomical shunt is called non-nutritional flow. Because in this shunt, no exchange takes place between blood and tissues. Let me explain, what is this blood flow? Now, what happens is that the blood that flows through a terminal arteriole moves either through the true capillaries or through the vascular shunts. Flow through true capillaries is called nutritional flow. It is called nutritional as it allows the exchange of nutrients between blood and tissues. When the precapillary sphincters are opened, they allow blood to flow through the true capillaries for tissue cell exchanges. On the other hand, when precapillary sphincters are closed, it restricts blood flow through true capillaries, and so it causes blood to flow through the vascular shunts. That is called non-nutritional flow, because here no nutritional exchange between tissues and blood takes place because of a thick vessel wall. And so blood bypasses the tissue cells with no nutritional exchange. Non-nutritional blood flow occurs in many tissues of the body, particularly during resting conditions or when metabolic activities are low, and there is no nutritional requirement for those particular tissues. So, what did we learn when non-nutritional blood flow occurs? Well, when the metabolic activity of those particular tissues are less, or in their resting phase when they don't need nutrients, or maybe other tissues required it more at that time. And where this non-nutritional flow occurs, it occurs through the vascular shunt or metarterioles, but not through true capillaries. And nutritional flow occurs only through true capillaries when tissues require those nutrients. Here, this amount of blood that enters a capillary bed through terminal arteriole is regulated by local chemical conditions, as well as arteriolar vasomotor nerve fibers. Based on body or body region conditions, a capillary bed may be nearly full with blood or bypassed almost totally. An example of such differing conditions involves eating. After a meal, the digestive process causes the blood to circulate freely through the true capillaries of the gastrointestinal organs, so that the breakdown products or metabolites may be received for absorption by the body. And obviously, we know it happens because the capillary sphincters are now opened. But as you approach the time of your next meal and are getting hungry, the majority of these capillary pathways are closed, thus only allowing blood to flow through the vascular shunt. So there will be no nutritional blood flow, but only non-nutritional flow. A different example involves vigorous exercise. Blood is rechanneled from the digestive organs to the skeletal muscles, because now skeletal muscles need more oxygen and nutrients than digestive organs. However, in the case of vigorous exercise after a meal, nutritional flow is now needed at both sites. That's why it confuses the body's management of blood to the capillaries and may end up causing abdominal problems such as cramping or indigestion. So, this was all about the general blood flow through the capillaries. So, we have learned in this section about different anatomical structures involved in microcirculation. And one thing that we learned here is that capillaries are a major part of microcirculation. But what are the features that makes only capillaries suitable for microcirculation? Or what features makes them a functional unit of blood circulation? Because, first, all other blood vessels are intact, and blood continues to pass through them. Only capillaries are permeable, and so contents can leak out of it. Second, capillaries have the largest surface area and thinnest wall, allowing more time for diffusion. Third, capillaries are present very close to the tissues, making exchange easy. So, these were all the features of capillaries, making them perfect for gaseous and material exchange between blood and tissues.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.